So this is another paper for ATP. Some animals that live in cold climate need to reduce the loss of heat from their bodies. Okay. And a student investigated the effect of using an insulating material on the heat loss of an animal. So for insulating, they have used something for insulating material, that is wool. Okay. And they have placed the test tube inside that beaker. And in one of the racks there is no insulation. So one has wool as an insulation, other doesn't. Okay. Immediately a timer was started and the temperature of the water in both test tube was measured. Suggest why both test tubes were filled with water only up to the marked line, not after, like it cannot be above that. Why? Okay, to ensure the same volume of water, first thing. Number two, to prevent the overflowing of water. Okay, because you have to add a thermometer later, so that may overflow and that may come out of the test tube. So its level should be a bit below to ensure same volume of water. Number one. uh, and number two, to prevent overflow when thermometer is added. Yes, because we need to check the temperature. How can we check the temperature without the thermometer? Okay. In a notebook, the student recorded the temperature of the water in each test tube every 2 minutes. And these are the values given. Okay. Now you have to make a table. Okay. Table is already given. Give the values for this. Okay. Where? Without insulation or with insulation? So we'll write here 73.0. Don't forget to put the decimal place, please. That is very important. Okay. 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 So what is it after 12 minutes? Read the values and then tell me. Let me make it bigger. Okay. 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 With insulation is 59 and it's 46.5. Correct. Okay. Next one, use the resultant table 1.1 to describe the loss of heat from the test tube and the effect of insulation. So what have you observed from this table? There is less heat loss with insulation. Okay. 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 Test tube with insulation uh, shows less heat loss. Okay. Okay, wait. Okay, okay. The insulation prevents excessive heat loss. Okay, okay. This is point number two. What is point number three? There is a very much important point. No, we didn't write that. There is more heat loss with time in test tube with no insulation. Okay. Or we can also say the heat loss in the test tube with no insulation is more than the heat loss in test tube with insulation. Okay. Okay, next one. Design an investigation to carry out to discover the effect of different surroundings, water temperatures on the loss of heat by the humpback whale. Use a test tube filled with water to represent a humpback whale. Yes, yes. So basically, you have to take a test tube instead of humpback whale. Hmm. No, no, they have written use a test tube filled with water to represent the humpback whale. So you have to use the same test tube like they have used in the start on the loss of heat by the humpback whale. Okay, water would be placed in what? Good, good, good. Mm-hmm. Constant. Okay, the initial volume and the initial temperature would be same for everyone, for each test tube. Good. Okay, okay, okay. We are calculating only temperature, so there is no need to take the final value of temperature. Take the final value of temperature only, not the water, sorry. Okay, not water. Okay, now you have to take at least three different external temperatures. How you would maintain that external temperatures? Good. Hot or cold water bath. Okay? Okay. Yes, yes, yes. And the time should be same. Okay. And what would you write in the end? How you would compare the result? Compare the heat loss for each external temperature. Okay. Compare the temperature or heat loss for each external temperature, like the hot water or cold water. Okay. The next question is okay, oximeter is given. You have to write your value over here. What is heartbeat rate and what is oxygen percentage? Okay. And heartbeat? 64. Okay. Now, we are not putting decimal over here as it's not mentioned here. Okay? Just keep this thing in mind. Okay. We have recorded the readings. Number two, you have to plot a graph. That is, I guess, a line graph. So, join the points with straight ruler. Okay? Don't make it with your hand. There shouldn't be curved. There should be straight lines. Okay? Okay. Suggest why it would not be valid to use a graph to determine the heart rate at 5 minutes. Okay. Okay, look over here. Between 2 till 7, the mid values are missing, right? 2 till 7, the mid values are missing. After 2 minutes, they directly jump to 7 minutes. So they are saying why it is not valid to give value to determine the heartbeat rate at 5 minutes. Data is not given. Data is missing. Okay? Data is missing between 2 to 7 minutes. Data is, okay, it's of 2 mark. Data is missing between 2 to 7 minutes. Don't write between like this, write a complete spelling, okay? 2 to 7, 2 and 7 minutes, okay. Moreover, okay, what would be your point number 2 since you have to use the graph? Point number 2, uh, okay, the change is not linear. When you draw the line, the change is not linear. You can only determine when it's increasing only or it's decreasing only. Okay, so the change is not linear. Oh, I have to write not. The change is not linear as well. Okay, I'll tell you. So if it's a straight line, you can easily determine. But if the line goes like this and this and this and this, you cannot determine it. So it's not linear. That's why you cannot determine it. Okay? Describe any change in the heart rate and the percentage of oxygen during the investigation using the data in graph. What are you observing from this table? What is the effect on heartbeat rate and what is the effect? Okay. Okay. The heartbeat fluctuates more. Okay. 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 Now let's go to the question again. Let's read it completely. Uh, yes. Yes. So, okay. Let's move to the values. Okay. So the oxygen doesn't change as much. There is lesser change in the percentage blood oxygen, while in the first 2 minutes, in the first 7 minutes, the heartbeat increases and then decreases after the 7 minutes. Okay, so this would give you 3 mark. Increases till 7 minutes and decreases after that. Okay, so we may say that the person may exercise till 7 minutes. Okay, then they exercise for 5 minutes. Okay, okay. At what time the value is coming back to its normal? Yes, heartbeat rate after 10 minutes. So we can also write that after 10 minutes the heart rate come back to its normal. Yes. Okay. Let's move to the next question. Okay. The student thought that the changes in breathing could have affected the percentage of oxygen in the blood. So just two changes in the breathing could be measured to help explain the percentage oxygen level during the investigation. Okay. Okay. What else can we use to explain the percentage? Okay. It's related to breathing. Wait, wait, wait. The terms are related to breathing. One is breathing rate. They have used the word percentage of oxygen. We can also write breathing rate, just like we have written heart rate. Have written heart rate. So we can write breathing rate. Okay. They are saying suggest two changes. One is breathing rate. Okay. Second is how deeply that person is inhaling. Breathing depth. So it's okay. Wait, 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 wait. Give me a minute. Let me write that. I'll explain. Okay. The thing is that they are only calculating the oxygen percentage through the oximeter. You can also calculate like the increase or decrease in the breathing rate when the person is exercising, right? So you can calculate the breathing rate, how many times he breathe during exercise or after exercise, right? Second thing, we can also measure or we can also observe the breathing depth, how deeply he is breathing in or out. Okay, they are saying suggest two changes that could be done. So instead of calculating the percentage oxygen, we can also calculate the breathing rate or breathing depth. these are the suggestions or the changes that we can make to make the experiment or the values more reliable or more accurate. Okay, when a seed germinates, this is about seed germination, a student decided to investigate the growth of radicle as the seed germinated. Okay, the growth is given. If you look at the marking, it is regularly marked over here. But after 2 days, the marking is different. There is increase in distance and some marking, and while there is no increase in the fume. Okay, describe the observable change. What have you observed? Just to observe which part is growing more. Okay, longer. Okay, there is uneven growth. If you look at these spaced lines, there is uneven growth. Some lines have grown more as compared to the others. Uneven growth in the spaced lines. Okay, suggest why the student placed the seed with the radical pointing downwards. Good. So that first of all it shows the positive tropism. uh, what was I going to write? So that the radical grow straight towards gravity, straight towards gravity. Okay, state two environmental conditions that should control to ensure maximum growth. Okay, what can be like given for the maximum growth? Temperature. Good. Next one. Okay, these are the chromosomes given. Okay, you have to label. Sorry, draw exactly. You have to draw only this cell. Calculate. No. Yes, yes. We have to make like this. A bigger size than this. So it should be bigger. Okay. And then put the correct numbers. Like it should be 2, it should be 1, 4, and like this. Okay. It should be like this. There is another one. Don't make 2 lines. I'm making 2 lines, but don't make 2 lines. Okay. Okay, next one. You have to measure the length and calculate the actual length. What is the formula of actual length? Image size. Learn this formula, okay? Are you sure? Okay, we are done with paper. There is no plan and investigation. Oh, okay. Okay, now you are free to go then.